Hi there fellow primates, welcome to this video. I hope it finds you well. Evolutionary flab. Mother Nature, she doesn't like it. She seems to follow the motto, use it or lose it. And what do I mean by that? Well, any animal body plans that have evolved adaptive appurtenances that are no longer contributing to increasing reproductive fitness, they degenerate, they get lost. So let me give some examples. Look for instance at this beautiful drawing from 1916 of a marbled sea snake. You would be hard pressed to find a more toothless snake than this. Evolving from venomous ancestors that use their venom to subdue prey, this snake in evolution has decided to specialize on eating mushy fish eggs. And for a diet like that, you don't need a venom apparatus. So if you look indeed at the skull of this sea snake, you can see that the venom fangs that once delivered venom have degenerated. If you would look at its venom glands, you can see they have shrunk as well. And even when you look at the venom in a molecular way, you can see that it has accumulated, the venom toxins have accumulated mutations that decrease their functionality, including debilitating mutations in the main paralytic neurotoxin of this snake. So if you don't use a weapon, you lose it. And that doesn't only happen in snakes. For instance, consider this peculiar animal. This is a bit of an unflattering photo, but I found it irresistible. This, of course, is an echidna, a monotreme just like the platypus. And it's not just peculiar because it has a four-headed penis. It's peculiar also because it can't get it up. Well, not the penis, I mean, but the venom spurs. Like platypuses, echidnas have a crural venom system, and this is a system of glands on the hind legs which end in a spur that you can see here on this picture on a stuffed echidna. And the platypuses use these spurs in fighting. The males use it to try to stab each other in fights for territory and for females. The echidnas don't do that because they cannot erect their spurs any longer. They can't cock the spur and put it into place because the bone structures have changed. So the echidnas cannot stab with their spurs. And also when you look at the molecular composition of echidna venom, you can see that very few genes that are actually expressed in platypus venom are expressed in the remnants of the venom system of the echidna, and also at much lower levels. What it does seem to function in perhaps is in scent communication between echidnas. So echidnas have evolved from venomous ancestors once shared with platypuses, but they have lost their venom system because they supposedly no longer need it. Perhaps because I wanted to speculate, perhaps because they have evolved a spiny covering and that is sufficient protection against enemies, but that is just arm-waving speculation. Also wanted to show you this cute little thing. This almost looks like a CGI creature that is being created for a movie, but it isn't. This is a baby echidna. If you think the baby platypuses are cute, look at this thing. And it has a cute little name as well. Baby echidnas are called, anyone? They're called puggles. This is a little puggle. It is fucking adorable. So echidnas and snakes in this case illustrate the use it or lose it motto according to which Mother Nature seems to operate. If you have expensive machinery that is expensive to build and expensive to run, which venom systems are, and you don't use them anymore, you don't need them to maintain reproductive fitness, you lose them, they degenerate. But what if a venom system doesn't want to be lost? In fact, what if the venom system thinks, I don't want to be in this body plan anymore. I'm sick and tired of it. I want one with better specs. I want to upgrade. I want to pimp my ride. What then? Well, what then is, check this group of creatures out. They're called Mixozoans, which translates into slimy or mucousy animals. And Mixozoans are parasitic creatures that need two different hosts to survive in their life cycle. An invertebrate host like an annelid worm or a bryozoan, and vertebrates hosts, aquatic vertebrates, often mostly fish. Okay, Ronald, where in this scheme are these creatures then? Where are the Mixozoans? Well, you can see in this scheme that there is two types of spores illustrated. Myxospores on the right and actinospores on the right, on the left. And these actually are the only free living stages of Mixozoans, because Mixozoans, when I started studying biology in the early 1990s, were considered to be protists, but Mixozoans turned out to be multicellular animals. However, unbelievably degenerated to this minimal body plan. And what they are actually, and this is based upon research over the last few decades, is highly derived endoparasitic cnidarians. If you look at this phylogenetic tree, you can see at the top the familiar entozoa. These are the corals and the sea anemones and their close relatives. And at the bottom you see a group called medusozoa, and this groups the jellyfish together, box jellyfish, hydra, siphonophores, the cnidarians that have at some point in their life cycle stage a jellyfish-like, medusa-like stage as well. And then in a clade related to that, to the medusozoans, you can see the Mixozoans. So Mixozoans already 600 or so million years ago diverged from the last common ancestor they share with medusozoans. So they are highly derived cnidarians. How highly derived? Well, essentially they have lost the entire body plan except the venom delivery apparatus. So in this schematic drawing, you can see on the left a typical stinging capsule and a metacyst from a cnidarian. In the middle you see a polar capsule from a Mixozoan, which is homologous to the stinging capsules of free-living cnidarians. And you can see that they have a lot of components in common. They're undoubtedly homologous. They are an intracellular organelle. Within them they have this tightly coiled up filament or tube, and then they can evert explosively in one of the fastest movements in the animal kingdom. When that happens to an metacyst of a cnidarian, they use it to deter predators or to kill prey. And in the case of Mixozoans, they do something else with it. It's those multicellular spores of the Mixozoans that have these organelles, and they use them to attach to and anchor to hosts, invertebrate hosts and vertebrate hosts. And then when they're attached with these spores, with these modified stinging organelles, they can penetrate the tissues of their hosts and become endoparasites. Now, I wanted to point out two things that are common to these as well. The capsule itself, the structural components of it are very similar between the Mixozoans and the free-living cnidarians, but also the components within the capsule that function in driving the expulsion of the nematocyst through an osmotic mechanism. The entire capsule is filled with positively charged ions, often of metals, and a negatively charged matrix of a biopolymer called polygamma glutamate, and we will come back to that in a second. So what you have here is essentially a venom system that has shed the entirety of the cnidarian body plan and thought, I'm going to pimp my ride. I'm going to ride around in better animals, superior animals, bilaterally symmetrical animals such as bryozoans, such as polychaetes, such as fish, amphibians, birds, and some mammals even, I think moles have been shown to have Mixozoan parasites. Now, one of the questions that remained after showing that such extreme parasitism and body plan reduction is possible in cnidarians is, well, does this retained venom structure, does it retain its bullets? Like free-living cnidarians, can Mixozoans actually shoot toxins into the hosts they enter as parasites? And I just wanted to highlight a few papers, including two from last year, that sketch an answer to this question. First benchmark paper was published in 2015 by Jonathan Fuchs et al. And it did the first transcriptomic study of a species of Mixozoan to find out, do they have homologs of toxin molecules you would find in the venom of cnidarians. And they studied a species of Mixozoan called Myxobolus pendula. If you translate it into English, it would be the hanging slimy lump, which is an untasteful sounding creature. And indeed, they found about half a dozen of sequences in the transcriptome of this species that looked like venom components of free-living cnidarians. Then 2 years later, in 2017, Gadi, Peryantinsky et al. Published the first proteome study of proteins secreted by a species of myxozoan called Overshasta. And they found actually that the proteome showed mostly structural components, the ones that built the stinging capsule, but none that corresponded to secreted toxin-like molecules. But as a benchmark first proteomic study of a myxozoan, it still gave us insights into the homology of myxozoan stinging capsules and also free-living cnidarians. And one thing they did find is, and that's shown here in this particular photo, if you look at the stinging capsules of myxozoans, you see here in dark blue illustrated that they're full of negatively charged molecules, and that the authors think could well be the polygamma glutamate that is involved in building up osmotic pressure, influx of water, and that expunges the filament and leads to nematocyst discharge. They also found indications that this creature contains enzymes that are probably involved in the biosynthesis of polygamma glutamate. So they may have that in common indeed with the nematocysts of free-living cnidarians as well. Now, a third paper was published by Eras, Spearer et al. In 2018, and that is the first genome level analysis of myxozoans. And what that found is that myxozoans indeed do have not only genes in common with the stinging cells of cnidarians, they have genes in common with them that are not found anywhere else in the animal kingdom. So really cementing the common evolutionary origin, the homology of stinging capsules in both free-living cnidarians and these highly derived myxozoan parasites. And then I come to the two last papers, and they were both published last year. One was led by a postdoc who was working in the Natural History Museum, still is, where I work as well, Ashley Hartigan, and it was supervised in amongst other people by Beth Okamura, another colleague and friend of mine at the museum. And if you don't know Ashley, I have a nice picture from December 2019. Ashley is standing in the front, holding a beer, wearing a white sweater. And this was one of the several Brexit parties that we threw, not because we like Brexit, we think it is a ridiculous idea, a damaging idea, a stupid idea, but you know, any excuse to give a party to get people together in your kitchen to eat, drink and converse. So hey Ashley, I think Ashley is at the moment somewhere in Australia in the desert collecting frogs for other work that she's doing. Long story short, she did the first analyses with her colleagues, combining transcriptomic techniques with proteomic techniques on a variety of species of myxozoans, 3 in total, and then comparing them to free-living cnidarians to see if there is any similarities and differences in the venom toxin-like genes they express. And what they find was very interesting. What they found is that, yes, if you look transcriptomically and proteomically, myxozoans have some venom components that seem to be conserved between them and free-living cnidarians, but far, far, far fewer of them than in the free-living cnidarians. A very streamlined, very reduced number of venom components that they may secrete. Now, to give you the actual numbers, for 3 species of myxozoans, one had 124 different sequences that match venom-like components, but in the proteome only 4 were found. So not all of these transcripts were translated into proteins. And then for a combined transcriptome of 2 other species of myxozoans, they found 96 transcripts coding for venom-like components, but only 2 of those were confirmed in the proteome. And as you can see here in the pie chart, one of those, and that is in the lower left here in the purple piece of the pie chart, these are serine proteases. These are in fact homologs of Blarina toxin. And if you've seen my video from last week about venom systems in mammals, these particular serine proteases, the callicrenes, were the hero of that story. So if you're interested in that, you can consult that video. And then a paper that was published in December, last December, just a month or so ago, by Benjamin Americas et al. And they had another look at certain Overshasta, the same species that was studied a few years before by Periantiskii et al. And they looked at transcriptomes, but they did something innovative, because the Mixozoans are parasites. They looked at little rainbow trouts that were exposed to this parasite. This parasite then infected them, and then they looked 1 day after exposure, a week after exposure, 2 weeks after exposure, and 3 weeks after exposure. So they made transcriptomes after 1 day of the gill, which was the area where the parasite enters the fish, and then 1, 2, and 3 weeks they looked at the gut tissue of the fish to look for Mixozoan specific genes expressed in these fish tissues. And they found a whole bunch. They found, in fact, 8 venom toxin-like proteins expressed by this species of Mixozoan that match 7 that were identified before by Ashley Hartigan and her colleagues, as well as the 2015 study by Jonathan Fuchs. And the thing that is outstanding in this paper, other than looking transcriptomically at the expression of these genes, is they made a time series. If you look at the expression of these different proteins, and you can see that in the schedule here, you can see that some are not expressed early on, like the one at the top, and only gradually increase in expression levels during later stages. And then others, for instance, the one near in the middle called hyaluronidase-like transcript, that is ramped up the expression early in the infection and then stays on a high level. And they actually have very interesting functional speculations, which is what they are, for what these particular toxin-like proteins may do in the life of these Mixozoans. So for the hyaluronidase, they speculate that it perhaps works to provide the parasite with access into connective tissues to enable it to enter the host and spread through the tissues of the host. Why? Well, many animal venoms express this particular enzyme, hyaluronidase, and it works for them as a spreading factor, opening up the extracellular matrix of tissues so that the toxins in the venom can spread from the envenomation site. And in this case, perhaps this Mixozoan uses the same enzyme so it can actually bodily infiltrate the tissues of the host. All these papers have, of course, many more details, so I urge you to read them. They're very interesting. And it gives you some tantalizing hints into how these creatures could have evolved and how they function, and how they repurposed what was ancestrally a predatory venom, probably mostly predatory venom, into a venom that is now used to infect and spread to the tissues of hosts. So yes, Mixozoans have totally pinned their ride. They were clearly not happy just living inside this inferior cnidarian body plan. They wanted something that was much more superior bilaterian body plan. But how? How could this have evolved? The unbelievable reduction in body plan and the specializations needed for a cnidarian to live inside not one but several hosts within a certain life cycle, extraordinary changes must have happened. And all you can do is fantasize about them, but Americas et al. Said there are some clues. There is what they call a phylogenetic and evolutionary intermediate between free-living cnidarians and these highly specialized Mixozoans that perhaps gives us a snapshot of an intermediate stage. And if you look again at the phylogenetic relationships of these creatures, you can see that the Mixozoans aren't just nested in a clade with the Medusozoans, the cnidarians with jellyfish-like stages in their life cycles. They are in fact the closest relatives of a thing called Polypodium. And this is a group with only one species called Polypodium hydriforme, which is a bizarre evolutionary cousin even of the Mixozoans. Now, although Americas et al. Call this a phylogenetic intermediate between free-living cnidarians and Mixozoans, it isn't. It is just the closest relative of Mixozoans, but still, when you look at the life cycle and morphology of these beasts, you can evoke before your mind's eye some sense of what may have happened on the evolutionary road between free-living creatures and these intra-tissue parasites, the Mixozoans. And if you look at this particular photo, you can see what Polypodium looks like. And it is weird. So if you look at A, you can see basically a chain of individual Medusa-like creatures that you can see dissociated in B. So A, this particular structure is called a stolon. It's basically a chain of individuals, and it's free living. This hatches from a fish egg, and I will get to that in a second, and then the stolon dissociates into little individuals that look like a more or less normal medusiform cnidarian. They have little tentacles, they have stinging cells and stinging organelles, and they hunt little invertebrates. They have a little mouth and a little gut. But these creatures at some point infect fish, specifically acipenseriforms. You go, oh, acipenseriforms, you mean paddlefish and sturgeons. Yes, that's what I mean. And they become an intracellular, intra-oocyte egg cell parasite of these fishes. A little larva develops inside the fish egg, and it's an inside-out creature. The gut lining, the little gastrodermis, is on the outside of the larva, slurping up all the nutrients from the egg, whereas its own skin, its ectoderm, is on the inside. Then it develops into this larger larva-like creature, into a stolon-like creature like you see in A, and at some point when it's ready to hatch, it inverts its entire body plan, adopting what is a normal organization for a creature, with the cells of the gut on the inside and the skin on the outside, in the process scooping up all the yolk from the fish egg, which it can digest at leisure. It hatches from the fish egg, and then you get this weird creature that starts to live a free living existence in fresh water. Absolutely bizarre. It is still of course, a venom system, then nematocysts, that is embedded in the cnidarian body plan, so it hasn't really yet pimped its ride. But that could happen in evolution. Maybe they are on their way to become as specialized and as unique as Mixozones. But if they have that ambition, they better be quick, because the acipenseriforms, the paddlefish, and the sturgeons, they are not doing well on Earth because of pollution and habitat destruction and overfishing. So it's not clear if they will have enough evolutionary time left, these Polypodium parasites, to survive long enough to become true specialized experts. Before I forget though, I want to go back. What does this mean for the venom? Do we know something about the venom? Yes, we do. And we go back to Ashley Hartigan's paper. And here is Ashley in another photograph. She's standing there on the right in the background smiling. This was last month when we had a little Christmas drink with some colleagues and friends. And Ashley and her colleagues found in the paper that if you look at the venom toxin genes expressed by Polypodium, they fall somewhere in terms of the number in between the very streamlined venom repertoire of the Mixozoans and a much broader venom repertoire of free-living cnidarians. And that makes sort of sense, because Polypodium has a life cycle with both a free-living predatory stage that uses its venom to catch prey, and this weird parasitic stage where it may use its nematocysts to anchor and board the host. We don't actually know yet how that process works, whether it is similar to what happens in Mixozoans, so there's plenty of scope for research here. But what we have here is possibly, in the mind's eye, some sort of conceivable intermediate stage on the road to extreme parasitism. Now, if you want to give these creatures a better chance to become even more specialized parasites before their hosts die out, I don't know, maybe you can help them. Maybe you can eat caviar infected with this particular parasite, and maybe you can give them a more high-spec ride. I don't recommend that. That's just a joke. Anyway, talk for 24 minutes, long enough. I hope you're all doing well. I hope you enjoyed this. If so, like it, subscribe, sign up for alerts, and I see you next time. Bye-bye.